Good morning. Today we'll be looking at the topic inhibitors of replication, transcription, and translation. We will first look at the overview of these processes and then go into studying the inhibitors of these processes. So, firstly, talking about replication, DNA replication is defined as the process of producing two identical copies of DNA from one original DNA molecule. DNA replication is often said to be semi conservative, meaning that each strand in the DNA acts as a template for the synthesis of a new complementary strand. Semi conservative because once the new DNA molecule is synthesized, it contains one strand from the parent and the other strand is a newly synthesized strand. DNA replication starts by taking one DNA molecule and giving two daughter molecules, with each newly synthesized molecule containing one new and one old parental strand. Now that we know the overview of replication, we will look at what are the inhibitors of replication. So, inhibitors are those molecules, those compounds that inhibit the process of replication. Quinolines are a key group of antibiotics that interfere with DNA synthesis by inhibiting topoisomerase, most frequently topoisomerase 2, which is also called as DNA gyrase, an enzyme involved in DNA replication. DNA gyrase, it relaxes the supercoiled DNA molecules and initiates transient breakages and rejoins phosphodiester bonds in superhelical turns of closed circular DNA. This allows the DNA strand to be replicated by DNA or RNA polymerases. So DNA gyrases plays a very important role and quinolines inhibit or stop these DNA gyrases from playing their role. The fluoroquinolines, second generation quinolines that include levofloxacin, norfloxacin, and ciprofloxacin are active against both gram negative and gram positive bacteria. Topoisomerase are present in both prokaryotic as well as eukaryotic cells, but the quinolines are specific inhibitors of bacterial topoisomerase 2. Rifampicin blocks initiation of RNA synthesis by specifically binding bacterial RNA polymerase. It does not interact with mammalian RNA polymerases, making it specific for gram positive bacteria and some gram negative bacteria. Some antibiotics that interfere with RNA synthesis by inhibiting RNA polymerase, such as actinomycin D, are not specific for bacteria and interfere with both mammalian as well as bacterial systems. These are most often used as antioplastic and anti-tumor drugs attacking rapidly growing malignant cells as well as normal cells. However, anti-tumor drugs cannot differentiate between malignant cells and fast-dividing normal cells such as those of the intestinal epithelial or hair follicles. Now moving on to transcription. What is transcription? Transcription is the first step in gene expression. It involves copying a gene's DNA sequence to make an RNA molecule. Transcription is performed by enzymes which are called as RNA polymerases, which links nucleotides to form an RNA strand using DNA strand as a template. Transcription has three stages. It is done through initiation, through elongation, and finally termination. In eukaryotes, RNA molecules must be processed after transcription. They are spliced and they have a 5 prime cap and poly A tail that are put on their ends. Now we'll move on to look at the inhibitors of transcription. Rifampicin binds with beta subunit of prokaryotic RNA polymerase. It is an inhibitor of prokaryotic transcription initiation. It binds only to bacterial RNA polymerase but not to eukaryotic RNA polymerase. Therefore, it is a powerful drug for treatment of bacterial infections and it is also used for the treatment of tuberculosis and leprosy. Actinomycin D, antibiotic, it inhibits transcription in bacteria and eukaryotes. The planar portion of this molecule inserts into the double helical DNA between the successive GC base pairs, deforming the DNA. It inhibits RNA elongation by restricting RNA polymerase along the template. It is a concentration dependent action. Going to the next one, translation. Let's look at what is translation. Translation is the process by which genetic code contained within a messenger RNA molecule is decoded to produce a specific sequence of amino acids in a polypeptide chain. It occurs in the cytoplasm following DNA transcription. Translation has three stages, just like transcription. It goes with initiation, elongation, and termination. Looking at the inhibitors of translation, these inhibitors selectively inhibit bacterial protein synthesis. The selectivity is due to the differences between the bacterial and human ribosomal proteins, RNA, and associated enzymes. We know that bacteria has 70S ribosome, whereas mammalian cell, it has 80S ribosome. So firstly, the macrolides. The macrolides are a group of antibiotics with a large cyclic lactone structure to which one or more deoxy sugars are added. Macrolides bind irreversibly to a site on the 50S subunit of bacterial ribosome, inhibiting the translocation steps of protein synthesis. It also interferes with transpeptidation steps. A few examples are erythromycin, clarithromycin, and different MgO concentrations are used of these. Moving on to azithromycin, which is concentration phagocyte, lysosome can be 40 times higher than in blood, and they can enhance intracellular killing of bacteria by phagocytes. Inflammation allows for greater tissue penetration. Erythromycin also distributes well to all body fluids except the CSF. It is one of the few antibiotics that diffuses into prostatic fluid, and it also accumulates in macrophages. Clarithromycin, azithromycin are widely distributed in the tissues. Azithromycin concentrates in neutrophils, macrophages, and fibroblasts. The mechanism of action of these has a unique mechanism as it acts on the sigma subunit of RNA polymerase, thereby disrupting bacterial transcription, terminating the protein synthesis, and resulting in cell death in susceptible organisms. These are my references. Thank you.